All right, so let's get started with our Kahoot for chapter 6. Um, yeah, let's get going. All right, chapter 6 covers genetics. So, what is the term that refers to an organism's total sum of genetic material? All right, so this is the genome. And so many of us are used to considering it like, is this the just the genes? No, it's not just the genes because you will have genes, yes, that code for proteins, or genes that, yes, code for RNA, structural RNA, and things like that. But something that we have to kind of consider when we're talking about the genome is a few things. First of all, you have your chromosomes. In bacteria, hold on, this was joining us. In bacteria, the chromosomes are one circular chromosome, like literally just a circle. Okay. But in eukaryotes, we have multiple linear chromosomes. We usually see them depicted as X's, but remember that's only during mitosis that they are like that. And so, we'll have usually most of them are in pairs if they are sexual organisms, a maternal and a paternal version of each chromosome. For humans, we have 23 pairs. And so these will be our chromosomes, but you can also have extra chromosomal DNA. So this is when we say extra chromosomal, this means outside of the chromosome. Okay. And so this would be things like your mitochondrial DNA. They have their own DNA that's not coded for by your genome, it's in your genome technically. It's not coded for in your chromosomes. It's not coded for in your genes that came from, the chromosomes from mama and dada. They have their own chromosomes, again, in the mitochondria. And so only the egg carries the mitochondria, by the way. Sperm do not have mitochondria in them. And so that's why we consider it to be maternal mitochondrial DNA, FYI. So mitochondria have their own DNA. We also have the chloroplasts. They have their own DNA if you have a chloroplast. So this would be our algae and our plants only. And then we also have things like plasmids. Plasmids are extra circle of DNA, double-stranded DNA, and contain extra genes that often help the organism survive better in like things like antibiotic resistance and stuff like that will be in the extra chromosomal DNA in that plasmid format. So these are extra bits of DNA that are in our circle format, but none of these are in the chromosomes. And so all of this is your genome. All right. And so when we talk about genes, can there be genes in the mitochondrial DNA? Yes. Can there be genes in the plasmid DNA? Yes. But there's also other information in that DNA that isn't considered really genes. And these are things that can help determine which genes are expressed or not expressed when we're talking about the control of the expression of the genes. And we'll kind of talk about that as this stuff goes on. So let's go ahead on to the next question. The physical expression of a genotype. So you have a genotype and you have what you what you see as the result of the genotype. All right, so phenotype, physical expression, genotype, your actual genes. This harkens back to kind of what we saw in this picture here. It's talking about if you have the big R gene for red, or the little r gene for white. If you have the big R from the mom and a big R from the dad, then your phenotype, what you see expressed, is a red flower. If you have a big R gene from the mom and a little r gene from the dad, little r being white, then you might see your phenotype as pink. And so obviously in your genes, we don't have little r's and big r's or whatever, but it just depends on what kind of a gene that you might have from maternal or paternal situations. And that what is expressed is what we see as the phenotype. This is like what determines whether you have green eyes or brown eyes or whatever, brown hair or red hair or black hair, whatever that is, or how tall you are. What's expressed and what we see from the outside is called the phenotype. Whereas your genes might have, different versions of the genes that might combine sometimes, but sometimes some genes are expressed over others. When you have two different versions of a gene and one is favored for expression over the other one, we call that favored version the dominant one. And it's dominant because it's the one that's going to be chosen if it's present. And this gets very, very tricky. I'm going to have to cough. Excuse me. So, this gets very, very tricky in genetics because some genes can be expressed equally and combined. Some genes are going to be stronger than others, but you don't just have two versions of genes. You might have multiple versions possible, and some of them are stronger than others, and some of them are weaker than others, and it's stuff like that. And so, and then when the body chooses which one is expressed or not, it gets very complicated. But for our basic version of the course, especially because we're focusing the most on bacteria, bacteria don't have two versions of a gene. They have one chromosome. So whatever version of the gene that they have is the one that gets expressed. There's no dominant or recessive or anything like that. And so it keeps it a lot simpler for bacteria than it would for eukaryotes. Oh man, I'm getting real hot. All right, next, let's move on. All right, for DNA nucleotides, it has three major parts that make up a nucleotide. What are they? Please read your choices. Okay, so we have in DNA, importantly, it's always deoxyribose sugar. If the sugar is ribose, then we're talking about RNA nucleotides, and they are different, and the distinction is important that we understand that. So in DNA versus RNA as well. So let's compare the two. DNA, that's a D. I'm sorry if it looks like a P to you, but it's DNA. The sugar that we see is deoxyribose. In RNA, we see ribose. They both have, nitrogenous bases. They both have um, phosphates, okay? But the nitrogenous bases can be different as well. So in DNA, we have A, T, C, G. In RNA, we have A, U, C, G. So the U is different. DNA also, we can, like, how do I want to put this? The form or whatever. DNA also is usually double-stranded, whereas RNA is usually single-stranded. All right. DNA is what makes up our genes. DNA makes up the genome for all cells, period. If you're bacteria, you have DNA, double-stranded DNA. If you're eukaryote, any type of eukaryote, whether you are protozoa, fungi, anything, all eukaryotes have double-stranded DNA cell in their cells for their genomes, period. The only difference that we see is with the viruses. So the viruses can have different types. They can have double-stranded or single-stranded DNA or RNA, okay? And so those are our major differences. And what else do I want to say about it? I'm not sure. Those are our major differences. What do we use DNA for? I guess we could put purpose on it. Period. Your DNA is part of your genome, your genetics. For RNA in cells, again, this is for cells. In RNA, this is always a working copy of whatever was in the DNA, but it can come in three major forms. There are other forms. We're not going to talk about them much in this chapter. I mean, it talks about it in the textbook. Please, this is definitely one of those chapters where you don't want to go to the textbook if you don't have to, because there's a way more information about this in the textbooks than what I talk about in my lectures. I promise you that the lectures are way more watered down than what the textbook is for chapter 6. So, in this case, um, can you guys hear me okay, by the way? I hope you can hear me okay. My microphone image is like barely moving. So, anyways, there's mRNA, which is our messenger RNA. This makes a copy of the gene that we're going to turn into a protein. There's tRNA, which is transfer RNA, which is going to be used in translation. It basically reads our mRNA and then determines what amino acid has to be going, matching up for it. And then we have ribosomal RNA, which makes up the structure of the ribosomes, where the pairing of these two will happen inside of a ribosome. And so we'll watch a video about that too, because this chapter has a ton in it, and we definitely want to be sure we're covering that. Okay, so those are major differences between the DNA and the RNA. Definitely, if I were to ask you, list two differences between DNA and RNA, I can't stress enough the importance of these two here. A lot of people will lean on the double strand and the single strand. And I mean, you're not wrong, but viruses do have double-stranded RNA sometimes. And so that can be tricky. So if I were to ask you a difference between DNA and RNA, what's the difference between DNA and RNA? Really, we're the nucleic acids, and that's in the sugar and in the nitrogenous bases that are seen there. So we use U instead of T. What does the U represent? Uracil. And you do need to know what the A, T, C, G mean. All right, so A, T, C, G represent adenine. It should already sound familiar. What is adenine? It's a nitrogenous base. That's you don't need to be able to recognize the structures either. You're welcome. Okay. You don't need to be able to know that they're purines or pyrimidines. I'm not going to ask you that in my class. What you do need to know is that A stands for adenine. And we've already learned about adenine a little bit in the form of adenosine triphosphate, which many people got wrong on the test. Adenosine triphosphate literally is an adenine plus a ribose plus three phosphates. Phosphate, phosphate, phosphate. This right here is what we call a nucleotide. It's just got two extra phosphates. So ATP, or adenosine triphosphate, falls into the category of a nucleic acid, not a carbohydrate. It's not a carbohydrate. Ribose, which is part of our nucleic acid nucleotide, is a carbohydrate. But the fact that we have adenine and a phosphate on there makes it fall into the nucleic acid macromolecule category. Okay. And this was covered in chapter 2. A lot of us get confused because we're like, no, you use glucose to make it. Yeah, you do. But did you know that you can change one macromolecule to another one? Just because you start as a carbohydrate, you don't have to stay as one. And I hate to break it to you. Just because you're eating protein doesn't mean your body's not going to turn it into a carbohydrate. Because what do you think it does? Just say it. Just because you eat a thing doesn't mean it stays the thing. Just because you eat collagen doesn't mean it stays collagen in your body. Just because you get infusions with NAD plus does not mean it stays as NAD plus when it enters your body. Your body will turn it into whatever it needs at that moment. Just saying. Now, of course, if you're going to be turning protein into a carbohydrate, it takes energy to turn it into that. And so, are you going to be burning more calories if you eat protein than if you eat carbohydrate that can be used directly for energy? You're going to be burning, it's going to use more energy to use protein as an energy source. Yes. So FYI, yeah, it still is going to burn more calories and you'll still lose more weight if you're using protein metabolism wise. So if you guys want to talk about that, we will cover that more in chapter 10, the metabolism. If you have questions about it, feel free to ask. Okay, so T is thymine in DNA. Again, in RNA, this will be replaced by U, which is uracil. C is cytosine, and G is guanine. I'm getting so hot. I shouldn't have. We're good. I just gotta wait for the Tylenol to kick in. All right. So these are our basic, again, these are called nitrogenous bases. These are not nucleotides. These are the nitrogenous bases. What makes a nucleotide is a nitrogenous base and then that 5-carbon sugar and a phosphate. NB for nitrogenous base. Okay. All right. Moving on. Any questions so far? Okay. My mouse sensitivity is way higher on this computer than at work. Okay, DNA antiparallel strands of nucleotides are linked by what? How are the strands linked together? Go ahead and delete my drawings so we can talk about this. People emailing me. All right, so they're linked together by hydrogen bonds. Good job. We got that. We got the general idea that sorry, that our DNA strands are linked together by hydrogen bonds. So what I'm asking about in this question is if you have a DNA ladder, basically, and you have each of these rungs of the ladder represents nitrogenous bases, right? And that they are linked together. Bear with me. In these dots here by hydrogen bonds. Just to remind you guys, what's happening with electrons in hydrogen bonds? No is the answer. Okay. So there's no exchange of electrons. There's no hogging or anything in the actual hydrogen bonds. What really is happening is that somewhere a hydrogen is having its electrons hogged by usually an oxygen. And that makes the hydrogen have a positive polarity. And then it's attracted to usually an oxygen that has a negative polarity because it's hogging from some sort of hydrogen, usually, or some other molecule, electrons. Okay. So that's what's really happening in a hydrogen bond, but this is the hydrogen bond. What type of bond is this one? Covalent. What about this one over here? Covalent. Unequal sharing of electrons, but it's still sharing of electrons in those bonds. Okay. All right. So FYI, I'm going to erase that. So we can leave that up because this is going to get heavy really quickly. Okay. And if you are watching the chapter videos, then you probably already know that it's going to get heavy quickly. All right. So think of it this way. If we have what we really have in our backbone here, what I call the backbone, the sides of the ladder, right, is really just alternating sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. I want to put enough on here that I can have that. Okay. And I wish I could just select all of this. Oh, I can. Okay. Good, good, good. Oh no. Oh no, you're annoying. Okay. Okay. And then good. Okay, this is going to be way better. Just come up here. I'm going to slightly alter this just a little bit because on the bottom It's not quite sugar, phosphate, sugar, phosphate, sugar, phosphate. It's more like phosphate, sugar, phosphate, sugar, phosphate, sugar. Because this strand operates in this direction, and this strand operates in the other direction. And the direction is denoted by whatever carbon is at the end of the sugar molecule. So you guys remember in chemistry, like I'm drawing my sugars like this, but you can actually draw your sugars like a linear format. And I mean, I don't remember the name. I like the Fisher structure versus like the Lewis structures and things like that. I don't remember which one is which, and I'm not going to test you on which one is which, but remember that we will number our carbons based on kind of what they're bound to. Well, as it turns out, on one end of our molecule, we'll have the number five carbon at the end. And by the other end of the molecule, on our sugar molecule, we'll have the three carbon. This is why we call it 5 prime and 3 prime, based on the carbon molecule that is there. And the other one is oriented in the exact opposite way, where this is, oh no, the 5 carbon, which we call the 5 prime, and the 3 carbon. Opposite directions, that the opposite directions are why we call it anti-parallel. The backbones, which are made of the sugars and phosphates of the double-stranded DNA strands, are oriented in opposite directions. And I mean, I could show you a picture of it. Hold on real quick. Just real quick. Not this one, the other one. And because I could try to draw it, but I feel like if I bring it up here, like you can see it better in the slide. So that like quite literally what is happening, is there a better picture of it? No, this is the best one that I have. That it has to deal with the number of the carbon. And so on the image here, you see how they've numbered every carbon. This is one, this is carbon two, this is carbon three, this is carbon four, I mean, four, and this is carbon five. That fifth one is the one that we're talking about. You might be wondering what's happened here. This is going to be like an oxygen or something like that. Don't worry about like the exact chemical composition, but it's just based on the numbering of the carbon that down here is our fifth carbon on this one. And so they're oriented in opposite directions. And again, I'm not going to show you pictures on my tests ever and have you label a thing in the picture. No, I don't have the patience for that. And I don't need you guys to have the patience for that because you don't need to understand it at that level for what you're going to use it for. But have an idea that what it's talking about is the carbons in the sugars in the sugar phosphate backbone. So when I say sugar phosphate, the D's are the picture here. This is deoxyribose. Okay, that's what the sugar is. And so it's alternating sugar phosphate, sugar phosphate, sugar phosphate, sugar phosphate. So sugar phosphate, all the way up one way, and then etc. So based on how you're looking at that. Okay, so I'm gonna go back to our thing. This. Okay. All right. So I hope that that is clearer with that for you guys. That I know saying that it's 5 prime to 3 prime kind of feels a little bit like, okay, sure it is. But what it's talking about is that sugar phosphate backbone is oriented in opposite directions. All right. So that's cool. So sugar phosphate, and linked to each sugar is a nitrogenous base. That's what's really happening when we talk about our DNA structure. And that if this sugar is linked to an A and the next one to a T, and the next one to a C, and the next one to a G, and I don't know what comes after that. That's make it up. Who cares? That on the other side, we will have T, and again, this is linked by covalent bond to the sugar. This will be A. And then this one will be G. And how do I know it's G? Because A always binds to T and C always binds to G. Can that ever be messed up? Yes. That's what mutations are. And so that can be caused by sun damage or mutagens, which we'll learn a little bit about in chapter 8. G, G. And then how are those guys linked together? Doot, 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 by hydrogen bonds. That's what's really happening. And that, again, a sugar, a phosphate, and a nitrogenous base, that is one nucleotide. One nucleotide. That's DNA. All right. And when we talk about RNA, it's just one of those strands. And again, replacing the T with a U and replacing the sugar with ribose instead of deoxyribose. All right, any questions about this? This is the anti-parallel arrangement of DNA. This is going over what a nucleotide is. Any questions about the structure of DNA before we move on? Okay. True or false? DNA polymerase builds DNA in the 5' to 3' direction. All right, so this is true, and I'm glad that you guys got that. Definitely always 5' to 3', never the other way. So what happens is, and briefly, because I know that you guys have already watched the lecture, and we are going to watch a video on it really quickly. The videos aren't long, and so I feel like the videos do a better job than my drawing. But briefly for the drawing, again, we have our DNA, which is a double strand, and it during replication, which is making copies of DNA. So this is also very key that we understand this. That what do I want to erase some of this stuff over here? We'll get rid of this overview here. I'll leave the basic drawing. All right, so in replication, replication always means DNA to DNA, making copies of DNA. All right. Transcription is going to be making RNA from a DNA, template. So DNA to RNA is transcription. All right. And then translation is going to be RNA to protein. I can't with this. Sorry, this computer's harder to write on than my ones in class because I can't actually see the screen that I'm writing on. I have to just like intuit it. Anytime you want to click on that, there it goes. Protein. Okay. All right. So this is the gist of how this is going to work um, generally. And then when we talk about reverse transcription, we can talk about it a little bit in this chapter, but it's mostly covered in chapter 8, that reverse transcription is the reverse of transcription. It's using RNA to go back into DNA. Our cells can't do that. Our cells do not have the machinery to do that normally. Viruses come with codes to make that type of machinery, and only viruses. And so, your body can't turn RNA into DNA. It's not able to do that. Only viruses have that. And it's very specific how they work. And they need more than one type of a gene in order to do that. And the way that it works is a little bit specific. So, but anyways, this is the idea of how this is what we consider the central dogma of biology. Replication is DNA to DNA, transcription is DNA to RNA, and translation is RNA to protein. And that's how our cells work. Well, when we're making copies of DNA, we will open up our DNA. And remember, it's not as simple to just draw the DNA because remember, one of strand is oriented 5 to 3, and the other strand is oriented 3 to 5. They're opposite directions. So when I want to make a copy of the DNA, first things first, I have to use a primer. And always that primer is RNA. This is because the polymerase, DNA polymerase, that's going to make copies of the DNA, can't add nucleotides. It has to add it to something that already exists. And so we have to use RNA as a primer for that. That's enough detail for what we need to understand with what's going on for this, okay? It'll use RNA as a primer, then we can add the nucleotide. If it's an A, we'll add a T. If it's a C, we'll add a G, and so on and so forth. Again, going in the opposite direction. 5 prime, straight on in all the way through. Imagine that these are all matching up, okay? And we'll add each nucleotide. We keep doing that in this direction. The 5 prime to the 3 prime, adding to the onto the 3 prime end in the 5 prime to 3 prime direction, okay? This is, and then we can continue opening that strand up more and more and more. This is this continuous strand. We call it the leading strand. The other strand, as we open it up, we can add a little bit of a primer and then add a little bit of DNA to it. So this one was opened up earlier and we built this part of this strand, again, because we can only go in the 5 prime to the 3 prime direction. This strand has to be made in parts. And so again, as we open this up, another primer in the dark green and then the light green, the match to the other side, whatever. And this one's made in fragments. The one that's made in fragments is called the lagging strand. Because we can make it as the other side opens up, because we can only add nucleotides in the 5 prime to 3 prime direction. Okay. And so after you're done, we will then use another enzyme. Again, a separate one will replace the RNA with DNA. It's another kind of DNA polymerase. The RNA primers get replaced into DNA. And then a separate enzyme called DNA ligase ligates or seals together ligase seals the fragments. Those fragments have a special name named after the man that discovered them, which is Okazaki. Okazaki. So again, it's not the Okazaki strand. It's the lagging strand that is made in fragments called Okazaki fragments that we will then seal together using DNA ligase. And so the overall result of what started out as, again, this was just one double-stranded bit of DNA that we then opened up at the site is called the replication fork where we're opening it up. We can't just open up all of your DNA. It's too much to work with. So we will open it at the replication fork. So what happens is then now we will have 2 new double strands. We will have 1 and 1, and then orange and then the other one will be green. Does this make sense? So you see how we kept part of the old and we made half of it's old and half of it's new. We have two new double strands. We called the daughter strands, half old and half new. Using the old as the template to create the new, right? This is what makes DNA replication semi-conservative. That's what semi-conservative means. That we kept half of the original double-stranded DNA to use as a template to build the new strand. Okay, let's watch a video. I don't know if the audio is going to come across. Now that's making me wonder. I'm gonna play it and then you guys, please tell me if you don't hear the audio, then I will know that it's not worth it for me to play it. Okay. I want to watch it, but it's also, I don't know if you'll hear it. This is different, the wrong chapter. There we go. Okay. Can you guys hear that? Because it's making audio. Did you guys hear that? You can type it in chat if you need to. You don't have to open your microphone. Did you guys hear any of the audio when it started?
It was really quiet.
Because it is loud on my end. It's probably like trying to pick it up through my speakers. Hold on really quick. I know there's a way to. Hold on, just bear with me. I don't know. I know there's a way to change it, but I don't know. Video. Oh, I don't know. I don't want to mess with this too much. So I just am going to recommend that you guys watch the videos on your own because like I have the volume turned up all the way on mine. So if you're barely hearing it, it's probably picking up through my microphone. That's not going to be enough for you guys to do it. Let me see. I can turn on the closed caption on the video, but that's the best I'm going to be able to do for it. Move this here. We'll try it and see if it helps. Turn it all the way up too.
Begins at a sequence of nucleotides called the origin of replication. Helicase unwinds the double-stranded DNA helix, and single-strand binding proteins react with the single-stranded regions of the DNA and stabilize it. DNA polymerase III is the major enzyme involved in DNA replication. DNA polymerase III can only add a nucleotide to the 3' end of a pre-existing chain of nucleotides, and it cannot initiate a nucleotide chain. Therefore, an RNA polymerase, called a primase, constructs an RNA primer, a sequence of about 10 nucleotides, complementary to the parent DNA. DNA polymerase III can then add deoxyribonucleotides to synthesize the new complementary strand of DNA. Because the two parent strands of DNA are antiparallel, they are oriented in opposite directions and must therefore be elongated by different mechanisms. The leading strand elongates toward the replication fork by adding nucleotides continuously to its growing 3' end. In contrast, the lagging strand, which elongates away from the replication fork, is synthesized discontinuously as a series of short segments called Okazaki fragments. When the DNA polymerase III reaches the RNA primer on the lagging strand, it is replaced by DNA polymerase I. which removes the RNA and replaces it with DNA. DNA ligase then attaches and forms phosphodiester bonds. The DNA is further unwound, new primers are made, and DNA polymerase III jumps ahead to begin synthesizing another Okazaki fragment. For simplicity, DNA polymerase III has been depicted as separate units, one acting on the leading strand and the other acting on the lagging strand. The
All right, hold on. All right, so that pretty much sums that up. I'm really trying, guys. I'm really trying. All right. So that pretty much sums that up. That's good enough for our purposes. So we're going to go back to our Kahoot. Maybe I keep figuring which window it is. Where is it? Yes, okay. All right, so that's how DNA replication will work. I hope that the video helps, and these videos are posted on Canvas if you guys need want to watch them on Canvas. Any questions about DNA replication? Okay. All right, which enzyme connects Okazaki fragments together that are formed during DNA replication? Which enzyme joins them together or seals them? All right, so that's DNA ligase. DNA ligase is the enzyme that will be sealing them together. Um, yeah, I mean, yes. So let's go ahead and just move on after this because there's enough to cover that I don't want to get too caught up on it. All right. Next question. During transcription, which of the following? So remember, transcription is DNA to RNA. I mean, it's over here on the left if you need to see it. Okay, so RNA is produced during transcription. In the picture, you can see it's very, very similar to DNA replication. We're going to open up the DNA at a site called the promoter region. The promoter is where RNA polymerase will bind if we're going to make RNA. And then we're going to make RNA in the 3 prime, 5 prime to 3 prime direction. Same direction. It's always 5 to 3. It's always 5 to 3. It's always 5 to 3. So in this case, we're going to open up the DNA and we're going to start making the RNA. But we don't need to make RNA off of both of the strands. We only need to make the RNA off of one of the strands because RNA is just single-stranded. So we will use one side of the DNA called the template strand to make our RNA. Okay, so we use the template strand as the template to tell us which RNA nucleotide to add next. Again, if there was an A on the DNA, we'll add a U for the RNA. If there's a T for the DNA, we will add an A for the RNA. It matches up the exact same way. It's just we're using RNA nucleotides instead of DNA nucleotides. And this is how I remember the difference between these guys, okay? I don't know why this doesn't let me click. Okay, there it goes. Okay. So replication, we are replicating. We are making the same thing. Replication of DNA is to DNA. Transcription, we are rewriting in different handwriting, but it's still handwriting. Again, think of the DNA as being your blueprint of everything you could ever need to build. We don't need the whole DNA, we just need a part of it. So we're going to make a working copy of part of the DNA. That part that we're making the working copy of is a gene. So we make a working copy of that part of the DNA. And again, think of it as like you go into the Smithsonian Institution, you write that down the instructions to build whatever you want to build, a chair, for example, and you rewrite the instructions. It's still in writing. It's just instead of it being typed up in a big old book, now it's in your handwriting for the instructions. It's still writing, whereas translating it is working it into a different language. Now we're going to put it in a different language. What language is DNA in? Nucleotides. What language is RNA in? Nucleotides, nucleic acids. What language is protein in? Amino acids. So we're turning nucleotides into a different language. Amino acids, we have to translate it into a different language, the amino acid language, to make our protein. And so that's how I remember which one is which. You do you for whatever works for you, okay? But you do need to know the difference between what transcription is, DNA to RNA, and translation, RNA to protein, okay? So transcription, we are making RNA. All right, next question. A codon is three bases of what? All right. I'm actually pretty surprised by how many of us got this one wrong. I was really kind of thinking we had this one. Okay, so a codon is not three bases of amino acids. First of all, amino acids are not made up of bases. They're made up of amino acids. So it couldn't be amino acids. They code for amino acids, but that's not what the question was asking us. The question is asking us what a codon is three bases of. And so this requires us to understand a little bit more about the whole process. So in our DNA, put DNA in green here. We have, I don't know, whatever, you have your sequence of A, T, C, G, G, C, C, whatever, which then in RNA, if that's our template strand, our RNA, which we'll put in orange, will be U, A, G, C, C, G, G, the complement, right? Every three DNA nucleotides we call a triplet. Here's another triplet. Let me actually even this out. We'll put two A's. Who cares? Each of these is a triplet. In RNA, UAG, CCG, GUU. Each of these three is known as a codon. A codon is three nucleotides, three bases, nucleotides of RNA. So our answer would have been mRNA. Okay. And then the tRNA, which I don't know what color to put. Let's put it in red. This weird red color, which would match up to that as AUC. On our tRNA. We'll just draw it kind of like this. Okay. And that it's attached to one amino acid based on that. On our tRNA, what do you think this is called? It's called the anticodon. So codons are on messenger RNA. Anticodons are on tRNA. And if the anticodon matches to the codon on the mRNA, then that would lead us to bring in a certain amino acid during the process of translation. So I'm going to try another video. Did the other video work okay when I set it up with the other way? I don't know if it worked okay, if you guys could hear it. I'm not smart enough to make it work. Because the translation one I feel like is a little bit more helpful. Okay, good. So I'll just do it the same way. Okay, so am I ready to watch the translation video? Let's do one more question first and see if we go in further to it. Okay, it goes right into operons. I don't want to do this question yet. We'll come back to this. Well, you can try to answer. In what kind of operons do repressor proteins bind to operators? So we're just going to jump into this because I already clicked on it. And then I'm going to come back to translation. All right, so that is inducible operons. We'll talk about that soon. Okay, so there's a lot to say about operons. We're going to get there. Okay, but I want to talk about translation first because it is a process that we definitely didn't talk about much in the questions, and I want to play the video for it. So we're going to do it the same way that we did the other video. Um, I'm not going to play the transcription one. There is a video on transcription if you are interested about it. But I don't feel like it's as necessary to watch the transcription video as much as it is the translation video, okay? So let's watch translation because there's no way I can draw it. It's impossible for me to really draw how complicated it is. Okay, so let's watch that. Let me pause it and we will make it big and put on our closed captions. Done.
Translation is the synthesis of a protein from an mRNA template. This process involves several key molecules, including mRNA, the small and large subunits of the ribosome, tRNA, and finally, the release factor. The process is broken into 3 stages: initiation, elongation, and termination. Let's see the process in action. Eukaryotic mRNA, the substrate for translation, has a unique 3' end called the poly A tail. mRNA also contains codons that will encode for specific amino acids. A methylated cap is found at the 5' end. Translation initiation begins when the small subunit of the ribosome attaches to the cap and moves to the translation initiation site. tRNA is another key molecule. It contains an anticodon that is complementary to the mRNA codon to which it binds. The first mRNA codon is typically AUG. Attached to the end of the tRNA is the corresponding amino acid. Methionine corresponds to the AUG codon. The large subunit of the ribosome now binds to create the peptidyl, or P site, and the aminoacyl, or A site. The first tRNA occupies the P site. The second tRNA enters the A site and is complementary to the second mRNA codon. The methionine is then transferred to the A site amino acid. The first tRNA exits, the ribosome moves along the mRNA, and the next tRNA enters. These are the basic steps of elongation. As elongation continues, the growing peptide is continually transferred to the A site tRNA, the ribosome moves along the mRNA, and new tRNAs enter. When a stop codon is encountered in the A site, a release factor enters the A site and translation is terminated. When termination is reached, the ribosome dissociates and the newly formed protein is released.
Okay. That pretty much sums that up. Okay. Get back to our Kahoots. All right, so that's translation. So again, transcription, which is actually making the RNA copy, happens very similar process to replication, but we're only making a single strand of RNA. Transcription happens in 3 steps. Initiation, where we bind to the promoter for RNA polymerase to bind to and open up our DNA. And then elongation, where we are adding our nucleotides for the RNA. And then we will get to a sequence that will cause it to end, and that's the termination step. Translation also has the same 3 steps. Initiation, which is our binding of our ribosome onto the messenger RNA, and then bringing in our very first tRNA into the ribosome to match up our anticodons. And then elongation is adding on the next guy based on the next codon anticodon pair. It brings in a different amino acid. We link the 2 amino acids together by a peptide bond. And then we just shift the ribosome down and shift it down and shift it down and keep shifting until we reach a stop sequence. What is a stop codon? A stop codon is a codon that doesn't code for an amino acid. So in our lecture, we talk about the different codons that can code for different things. And we see there is some overlap. We see that, any CC, it doesn't matter what your third one is, codes for proline. Any AC, doesn't matter what your third one is. Any AC, X, whatever it is, ACU, ACC, ACA, or ACG will code for threonine. And then some of them we only have 2, and then other ones only have 1. So UGA is a stop codon, and so on and so forth. They're stop because they don't code for an amino acid. They are nonsense codons. A nonsense codon is a codon that does not code for an amino acid. And so that dictates building our protein as a result of all of that during the translation process. So again, to review all of that, we have our DNA triplets. We use one strand as our template to make our mRNA strand. Our codons match with our anticodons on our tRNA during translation. That will lead to 1 amino acid for each codon on our mRNA, again, matched up by the anticodon on our tRNA. And then that's how transcription and translation works. Do we have any questions about replication, transcription, or translation? Because I am going to go over the operons next, and it is a lot for people usually. All right. I'm getting rid of all these drawings. And we're going to go back to our question about the operons. I can literally hear my husband snoring in the other room. And I don't know, y'all can judge me if you want to, but the sound of snoring, not my own snoring, of course. I know this is probably hypocritical, but somebody else snoring, it's like nails on a chalkboard to me. And you guys probably can't hear it. You're blessed. And he is in a whole other side of the house, and I can hear it in here. He's sick too, bless his heart. So I don't want to go be mean. I'm just going to hold it in to let him rest. Okay, so in operons, operons. It's going to drive me insane though, the fact that I can hear it through my headphones across the house. Okay. Anytime you want to open. Here it goes. Okay. All right, so if you guys end up being able to hear it, just let me know and I'll go tap him on his little shoulder and let him know. If he lays on his other side, he will be quiet. It's like he's on his wrong side. So just let me know. Okay, so operons. An operon is only in eukaryotes, bacteria and archaea. So mostly when we talk about this, we're talking about bacteria. Archaea can have them too, but we're mostly talking about bacteria, okay? And these are a set of genes that are controlled by one promoter and then managed by one operator. Okay, so kind of controlled by both. Controlled by one promoter and operator. Let me draw it for you. This is in the DNA, not in the RNA, in the DNA. So in DNA, remember what a promoter region is. It's where your RNA polymerase will bind to make the mRNA. So in the DNA, we have the promoter sequence. Then we have an operator sequence, which I'll put as O. Okay, promoter, operator, and then our genes, gene 1, gene 2, and gene 3. In eukaryotes, all of our genes are controlled separately. We don't have this multi-gene linking thing going on, okay? Eukaryotes, each gene is controlled by its own promoter. This is only in bacteria that they have this. And so this is all genes that are related to a similar function, related to doing a similar function, a similar end goal. In this case, making an enzyme to break down lactose. We might need to express 3 genes that will work together to break down lactose. Lactose is a disaccharide of, I believe it's glucose and galactose. And so I might be wrong about that, but it is a disaccharide for sure. So it means it's 2 sugar molecules linked together that an enzyme is going to have to be built to break it down into usable forms. So in order to break down lactose, we don't need to make the enzymes all the time. We can turn it on and only make it when we need the enzyme. So this is how we control. Operons are how we control the expression of genes in bacteria. It's one way. There are 2 types of operons, repressible and inducible. We're going to start with inducible. If a woman is not in labor and you want her to be, so her labor is turned off and you want to turn it on, and you can induce the labor. So inducible operons are off and they can be turned on. Whereas repressible operons can be repressed, can be turned off. These guys are always on and can be turned off. Okay. How does this work? This is our gene setup. And to start off with, RNA polymerase usually always binds at the promoter. Again, opens up the DNA and we can make mRNA. That's normally how it would work. This is on. What I'm drawing right now is an operon that is on. Okay, so hold on, let me make a copy of this before I get too far. We'll just do the whole thing. Sure. No, what? What? It's not letting me make a copy of it. That's okay. Anyways, it's fine. I'll just redraw it. It's fun. We're having a good time, right? Okay. So again, this is our DNA. We have our P, our O, and our different genes that are downstream. Nails on a chalkboard in the next room. Not even the next room, literally on the other side of the house. I can hear it even through the headphones. Watch me spin out from it. Okay. So again, if you are on, maybe we do it in green because it's on. If you're on, RNA polymerase can bind to the promoter and then make mRNA from the DNA. Okay, this is what it looks like when it's on. Otherwise, if it's off, we have a repressor bound to the operator. In this case, RNA polymerase can't move forward. So in this case, our genes won't be expressed. We won't make mRNA. We won't be making it. Okay. And so this is off. Literally, it's just whether you start off or you start on, and you can switch between the two. If you start off, then you are inducible. You can be turned on. If you start on and you can be turned off, then you're repressible. So how do we remove the repressor? Because this is the key, right? That repressor and removing it, or that repressor and making it bind. Those are the key to whether you can induce or repress. So in order to make the repressor bind, in this case, if we're talking about the lactose operon, the way the lactose operon works is that you have a repressor that binds to something else called a co-repressor. In this case, this happens to be lactose. When it is bound, I shouldn't have done it in green. When it is bound, it's driving me insane. Repressor binds to, let's do purple, lactose. I don't know why it makes me think of purple, but it does. That co-repressor causes the repressor to bind. So when lactose is present, the repressor can bind to the operator. When lactose is absent, the repressor won't bind to the operator. It works the opposite way for our repressible operons. If it's just on, there will be a repressor hanging out somewhere. When we make too much of our end goal, our product, in the case of a repressible operon, the product is usually what binds to the repressor and activates it and causes it to bind on. Does that make sense? So you make too much of your product, it's not getting used up. If you're at a factory, for example, you have too many pairs, like too many pairs of pants that are being made. People aren't buying enough of the pants. You're not going to keep making the pants at your factory. You're going to say, oh, we have a buildup of pants. Let's stop making pants. It works that way with our repressible operons as well. In the case of the inducible operons, it's like it's saying, we got an order for pants. Let's make the pants now. We can't make the pants without the order. The order means we remove the repressor. And so that's the idea of how the operons work for the bacteria. Do we have questions about the operons? Please ask now if you do. Is this? Oh, it's a scratch on the monitor. Okay. All right. I hope that covered the operons okay. Are you going to get tested on operons? And I'll give you, maybe describe a scenario of, hey, we have an operon that is on and then it got turned off by its product. What type of operon is that? If it was on and it got turned off, then that would be a repressible operon. Questions like that will be on the Unit 2 exam. So if you end up studying and you're still confused about what type of operon is which, then please let me know when you get to that point. Okay, we're going to move on. Our last question. Missense mutations. What are they? Okay, so let me bring this up really quick because I'm going to need this. Because I don't have all of my amino acids memorized. So there's several different types of mutations. I don't want to tell you guys wrong. You do not need to memorize any of your codons or anything like that. I will always give you the sequence and then tell you what's going on. If your original DNA sequence was UUU, or not DNA, but your original mRNA made from your DNA template. So your DNA would be AAA. Your RNA is UUU. And it usually codes for, UUU codes for phenylalanine. If we changed that to UUA, that's just one nucleotide, right? That now codes for leucine. This is known as a missense. If you change the amino acid, you change the nucleotide that leads to a change in amino acid, then that's a missense mutation. What if we had changed it to UUC? Did we change the nucleotide? Yes, we changed the nucleotide, but it still codes for phenylalanine. Is that a mutation? Yes, the nucleotide changed. The DNA sequence changed, the mRNA changed, but did the resulting protein change? No. This is known as a silent mutation. If we had changed UUU instead to, I don't know if I have one that I can choose that's going to work for this example. I don't know all these. I don't know. Why? No. I just want you to be over here. Okay. Stop. I don't want that. Like, actually stop doing that. I don't want that. I don't want it. Stop doing it. No, down here. Okay. If you had changed it, and you can change more than one nucleotide, so just imagine that that is what is happening, okay? Oh my God. I'm gonna try not to crash out right now. I'm just not feeling well. Okay. If you had changed it to UAG, this would be in case we changed two nucleotides here, this would be a stop codon. This would be a nonsense. So if you change your original codon to a different codon that changes the amino acid, that's a missense. If you change it to a different codon, but the amino acid is the same, that's a silent mutation. If you change it to a different codon that changes it to a stop, then that is a nonsense. Let's imagine that you are reading something that is UUU, CCC, GGG, AAA, and whatever they code for. I don't know. We already know phenylalanine. Who cares what the rest of them are? Let's say this one is glutamate, and this one is aspartate, and this one is, I don't know what, leucine. Who cares? Okay. If this was your original. And then we changed it to where we remove this U. So now it's not UUX. It becomes UUC. Then CCG. Then GGA. Then AA, whatever comes next. Removing a nucleotide or adding a nucleotide is what we call a frame shift mutation. Imagine that you have a big book and you remove one letter but keep all of the words the same length. You just shift the letters over. So instead of the cat in the hat, take out the E, and now it's the k, ati, nt, he, a, whatever comes next. And now you can open up that book in the middle somewhere and you can't, doesn't make any sense anymore. That's the difference with a frame shift mutation. Everything just gets moved over to compensate for that lost nucleotide and whatever. So the most devastating ones that we're going to see are when you get an early stop or when you have a frame shift. These are often the most deadly types of mutations, not making our full protein or not, having an issue with that. So missense mutations, you're making a different amino acid than you're supposed to. Silent mutations, you still get the same amino acid, even though the nucleotide changed. Nonsense mutations, you're getting a stop where there should be an amino acid. Frame shift mutations, you're going to be shifting the entire reading frame downstream from wherever you have added or removed a nucleotide. And then one other mutation is a back mutation. And a back mutation is like if you had this UUC silent mutation, UUC, and then it changed into UUU again, the wild type, the original. So back mutations are going back to the original. We see mutations quite a lot in bacteria and even more in viruses. When those guys replicate, if they have a mutation that kills off the progeny cell, who cares? We're going to make millions more in a matter of, minutes. So it doesn't matter. So they can afford to have mutations because some of those mutations can lead to advantages in those bacteria or those viruses. For example, if you just leave E. coli out in the presence of an antibiotic, eventually you'll just get a random mutation that will allow it to survive on the antibiotic that used to kill it. Eventually it will happen if you allow it to, which is important that we take our antibiotics to kill off any of the susceptible guys so we don't have weird outliers coming through at any time. So that stuff is really important when we talk about mutagens. Mutagens are things that can cause mutations. Example of mutagens would be ultraviolet radiation, and there are many certain chemicals that can cause mutations like that. Okay, so that's it for chapter 6. Do we have any questions about chapter 6? Yes. Jackie, you can use this screenshot for your Monday, Wednesday Kahoot. This will be for chapter 6. So we're going to do chapter 8 next. So if you're joining for Monday, Wednesday, this can count for that. Okay, so we're just gonna jump into chapter 8, and then we might come back to the worksheets after this. But I definitely want to be sure we're getting the Kahoots in for chapter 6 and 8. And most of chapter 8, well, we'll just jump into it. I don't want to get too ahead of myself. Just double check that you're signed in for chapter 8. Okay, it should be everybody. It's really loud in my ear. It drowns out the snoring. I love him, but okay. Let's get started. So chapter 8 is going to be discussing how to use all of this ideology in science, like how we're going to take advantage of it. So what type of biological macromolecule are restriction endonucleases? So anytime you hear something called ACE, DNA ligase, um, DNA polymerase, restriction endonuclease, ACEs are our enzymes, and enzymes are always proteins. These are proteins that act on DNA. In the case of restriction endonucleases, They cut DNA at palindromic sequences. Specific palindromic sequences. I'm going to show some pictures. Again, it's going to be better than me drawing everything. So this time I do actually need the right one to pull up. Not CRISPR. There we go. All right, in the case of our restriction enzymes, okay, we have a sequence here, GAATTC. Let's pull it up. So if we draw GAATTC, what is the opposite of that? Again, if this is the 5' direction and this is the 3', what does the opposite direction say? If we go literally from the opposite direction, from the 5 to 3, we have GAATTC. Would you look at that? That it reads the same way from 5' to 3'. This is a palindrome. It reads the same backwards and forwards. Okay. And so what happens at these palindromic sequences is that these enzymes kind of clip it like this to create these kind of what we call a sticky end. Okay. So in this case, in the picture here, if this was our sequence, one of the strands would be G, and it would be stuck to the C at the other side, which would have an overhang of a T, T, A, whereas the other one would be broken off, where it would be have an overhang of an A, T, T attached to a C, which is, again, hydrogen bonded still to a G. And these are our sticky ends. So they come apart. They're sticky because this side wants to bind to its match on the other side, but we've been separated by this enzyme that cut them apart like this. How is this useful? Well, if I create a sequence that has AATT and then the gene for insulin, and then stick another AATT at the end, now I can insert this in between in here, which is what we are having in here. So you can take DNA sequence from one organism, pack on a sticky end on the end of it, and then stick it into the DNA of another organism. In this case, this would be a plasmid, a little bit of circular DNA, still double-stranded, that we can just insert that target gene into. Okay, so this is the idea of how we can do that. Now, with restriction enzymes, one enzyme, in this example that I'm using here, we're talking about EcoR1. This is an enzyme that comes from E. coli, restriction enzyme number 1. It cuts at this specific palindrome, no other place. HIND3, which comes from Haemophilus influenzae, or something, enzyme number 3, okay? That this cuts at that sequence. And then we have another enzyme that cuts at another sequence. And then a different enzyme that cuts at a different sequence. And that these are all different restriction enzymes that only cut at very specific sequences. Some of them create sticky ends, like we talked about. Other ones create blunt ends. The most useful ones, the ones we're going to talk about the most, the ones I'll definitely be testing you on, are the ones that cause the sticky ends. And you don't need to memorize which enzyme cuts where, because there are thousands. You're not going to memorize all, it would be impossible. But one enzyme only cuts at one place, and it's always a palindromic sequence, and so it's kind of limited at where it can cut. So restriction endonucleases, each specific nuclease only cuts at one palindromic sequence. We can design our genes to using our understanding of nucleic acids and things like that to have certain sticky ends, or we could use CRISPR. And CRISPR doesn't have to rely on palindromes. You can tell CRISPR to cut anywhere in the DNA. So CRISPR-Cas9 is a system that basically can recognize any sequence and cut it wherever you want. It doesn't rely on palindromes. It could technically cut at any part of your DNA. Okay, so CRISPR is like this, but without the palindromes, and you could technically tell CRISPR any gene where you want it to cut. Both of these systems, restriction endonucleases and CRISPR-Cas9, both of them work for bacteria. They're found in bacteria naturally to help eliminate foreign DNA they don't want in their cells, usually plasmids that they don't want there, or bacteriophage DNA that they don't want there. Normally, that's what these restriction endonucleases or CRISPR-Cas9 will normally allow the bacteria to remove unwanted foreign DNA from plasmids or from phages, right? So normally that's what they do. But as scientists, we're hijacking that. Okay. So let's move on. Let me erase this real quick. Okay. I'm going to leave this up for a second. All right, question 2, restriction endonucleases. What are they? I have it drawn up here, and the picture kind of tells you a little bit what they do. All right, so they are molecular scissors, and they only cut at specific palindromic sequences. Good job. So we're going to move on. True or false, restriction enzymes can cut anywhere in the DNA sequence. All right, so this is false. They can only cut at very specific palindromic sequences. And that little video that it had playing in the background is kind of an example of how kind of they will join things. It was playing, but now it's only just a picture. But this is a picture of those sticky ends. Okay, good. It did kind of go. I don't know why it doesn't just play. Yeah, it creates these sticky ends, and then they can be brought together and then be sealed together using DNA ligase. Again, anytime you have fragments of DNA being joined together, they must be sealed by DNA ligase, the same enzyme that sealed together our Okazaki fragments. I've got a cough. Okay, let's keep going. True or false, CRISPR-Cas9 is a system in bacteria that can be used in genetic engineering to cut just about any sequence. Now, is that true or false? Yeah, so this one is true. So CRISPR-Cas9 can cut anywhere, whereas the restriction endonucleases can only cut at specific palindromic sequences. So that's the beauty of CRISPR. We could tell CRISPR to cut anywhere in your DNA. Inserting genes into the cut sequence, though, so we can cut your DNA and remove genes, but what do we replace it with? And then replacing it is the problem that we have in actual practice in real life. We haven't been able to get this to work effectively in mammals. Take that as you will. Science says, anyways. And anybody that I know that's actually tried it in practice, they're not just being silenced by big pharma, I can assure you, because I know the people doing the work. Like it just isn't translating into multicellular organisms because how do you get it to every cell in the organism? So, one thing that can get genes to every cell in an organism is viruses. So you can use viruses to force viruses to insert certain types of DNA. That's not CRISPR. That's not CRISPR. Just FYI. Okay, that's viruses doing that. So, anyways, we'll talk about that in a little bit. That would be gene therapy. And that's coming up in this chapter. All right, moving on. PCR. What does PCR do? Don't worry, we'll talk about it. One of the biggest concepts in this chapter. PCR stands for polymerase chain reaction. And our answer here was the red one. It amplifies DNA in vitro. So this also requires us to have some understanding of what in vitro and in vivo means. Vivo means to live. Vitro is like in a test tube. Okay. So where are we actually amplifying the DNA when we do PCR? We're amplifying the DNA, making copies of it in a test tube in vitro, not in vivo, which is in the body. Most of the testing that most of our systems are going to rely on in testing for our illnesses or whatever in laboratories, in hospitals or whatever, when we do testing, it's usually ex vivo outside of the body and usually in vitro in like a test tube situation. Okay. So PCR, typically we are going to make DNA into DNA, which is replication. So it's the same exact process as DNA replication. But in PCR, we don't need to make a copy of the entire DNA. We only need to be sure we're making a copy of a target gene. And this relies on the use of a primer, and it's usually 15 to 30 nucleotides long. A, T, C, G. So A is a nucleotide. T would be representing nucleotide. What do they really represent? A nitrogenous base. But when we talk about it, we're talking about the sugar phosphate backbone with it. So a sugar, a phosphate, and a nitrogenous base is a nucleotide. Okay. And so this 30 nucleotides would represent a target that is unique to whatever you're testing for. So, to start, if you have a DNA sequence, whatever it looks like, here's your original DNA. I'm not going to draw 30 nucleotides for our thing, but if our primer binds to one end of it, go away. Again, it'll be what 15 to 30 nucleotides. If your primer binds, then you can match up the respecting nucleotides on the other side. The next one and the next, because remember, DNA polymerase can only add nucleotides to the matching side if you have a primer present to add it to. So if the primer binds, if primer I don't know why this is being so slow. It's really bad. Come on, dude. Just suddenly it's being janky. If the primer binds, then you can elongate. You can add nucleotides. It's really being laggy. Really laggy. Okay. So only if the primer binds. So it's all determined by if the primer binds. If the primer doesn't bind, you won't add nucleotides. So quite literally, if I am asking the question, does my patient have, are we going to do PCR? Does my patient have? Why is it so laggy just suddenly? It's been fine this whole time. Does the patient have? What do you mean? You've been fine this whole time. COVID. I have to write it slow, apparently, like slower than molasses, suddenly. Then we will use a primer that only matches COVID genes. If the primer binds, then you can add nucleotides, and the machine can detect if we're adding complementary nucleotides. So if my sample that came from my little nostril was put into a test tube and we heat it up to separate my DNA strands, we'll have two separate strands. This again is done by heat because we started off with our first step. We started off with our DNA double ladder. And we used heat to separate. When we separate a DNA strand like this using heat, this is called denaturing. Denature. Bear with me. I'm so sorry. This is going super slow, this lagginess of this. All right. That's the first step of PCR. That's the first step. Then we'll cool it just a little bit to anneal. That's the second step. Anneal means bind. The primer. So you could call this the anneal step, or you can call it the priming step. Both will be acceptable. Why is it so laggy? Okay, that's the second step. And then the third step, we'll do it in blue, sure, is the elongate step. I bet it's because my husband's on the internet and it's taking up internet bandwidth. So again, add a nucleotide, add a nucleotide, add a nucleotide, add a nucleotide, and whatever. And again, these processes will be happening on both strands. So the primer on the other side, again, remember 5' to 3', they're in opposite orientations. This is really what PCR would be doing. And it is, PCR is the molecular Xerox machine. It is making copies. Each round of PCR will make a copy of the existing nucleotides. Okay. So again, this is all one round, 3 steps every round. So once we get our, full copies made, we went from 1 strand to 2 strands, and that's 1 round. I wrote a whole word, but it's just not coming across for some reason. I don't know what's going on. Okay. All right, so that's how PCR works. It depends on, did the primer bind? If the primer bound, you can elongate. And as you add the nucleotide, it gives a little spark of color. If you add an A, it'll spark blue. If you add a T, it'll spark red. If you add a G, it'll spark yellow. If you add a C, then it'll spark pink. I don't know, whatever colors, okay? And then based on what color comes out, the machine can detect the colors that are being given off and saying, oh, we are elongating. If you keep elongating, if every time you run through a cycle, oh man, we keep having primer binding and we keep elongating, then you're positive for the target gene for COVID. It only tests for one thing though. So If you swab my nose and you want to test me right now for the flu, you would swab my nose, put it in a PCR, put it with primers that detect flu genes. What would happen then? The primer wouldn't bind and it wouldn't elongate. That means a negative PCR test. If you mix my nose swab right now with primers for COVID genes, the primers will bind and it will elongate, and that would be a positive PCR. It's that simple. It's way simpler than you probably thought it was. We do have PCR tests out there that can test for multiple things at once. They are extremely expensive, and telling the difference depends highly on the genetic makeup of the genes that you're testing for. And so that's way more complicated than we need to go into for this class. But if you want to have that discussion, we can. I'm not going to test you on PCR techniques that test for multiple things. But the problem is you need to know what primer is binding and what gene you're copying if you're testing for multiple things in a PCR test that tests for multiple things. That makes sense. Most PCR tests only test for one thing, and it entirely depends on the primer binding and elongates. If it elongates, you're positive. If it doesn't, the gene isn't there, you're negative. And it's that. So you have to have primers for the thing you're testing for, and you have to have the ability to detect if you're elongating. And that usually depends on having a program for the computer that's doing this to be able to detect whether it's elongating for each thing. So it can be specific. So this is why you can't just test for anything on a PCR machine. You can only test for what the PCR machine is programmed for and for the primers that you have available at that time. All right. So those are the 3 steps of PCR: denature, using heat, separating the DNA strands; anneal, causing the primer to bind to the target sequence by cooling it down slightly; and then elongate, having DNA polymerase add on the matching nucleotides. In each round of PCR, you will go from 1 strand in the first round becomes 2 strands. That's round 1. In round 2, each one becomes 2. So in round 2, you go from 2 to 4. Whereas in round 1, you went from 1 to 2. So you are just doubling every time. What will we have in round 3? 2. Where do I? Oh, it's fine. It's fine. Each one becomes 2. So 1, 2, 1, 2, 2, 1, 2. So in round 3, you went from 4 to 8. You don't start with the number that is the round. Does that make sense? So if you are all, if I were to say to you, you started off with 1 piece of DNA, how many pieces of DNA will you have after 4 rounds of PCR? How many would you have after 4 rounds of PCR? Does anybody know? Anybody want to make a guess? Type it in the chat, please. Type it in the chat. Type it in the chat. I'm gonna wait till I see at least one answer in the chat. I know y'all are paying attention. After 4 rounds of PCR, if you started with 1, after 4 rounds of PCR, how many pieces of DNA will you have? I've got you up to 3. If after 3, you would have 8, how many would you have after 4? This is a test question, by the way. It might not be 4. It might be up to 6. Good guess. We got 1 guess. 16 is correct. It doubles every time. It doubles whatever you had in the previous round. So after 1 round, 1 becomes 2. After 2 rounds, 2 becomes 4 after 3 rounds. 4 becomes 8 after 4 rounds. 8 becomes 16 after 5 rounds. 16 becomes 32 after 6 rounds. 32 becomes 64. I might ask up to 6 rounds. You should be able to do the math on your own, and you are always welcome to draw it out if you need to. Okay? So this is perfect. This is how this would work. Good job. We're getting there. Okay, and if this is confusing to you guys, we will have a chance to talk about this in Lab 10. Just while I'm talking about Lab 10, when we get to Lab 10, so our next lab on Thursday, Wednesday or Thursday, for Monday, Wednesday people, it'll be Wednesday tomorrow. And then Thursday will be Thursday. We'll be Lab 7. We're just skipping over Lab 6. We'll come back to it. Okay? Lab 7 is too long of a lab for me to double it up with something else. Okay? So 7 will be by itself on Wednesday and Thursday. Next Monday and Tuesday will be Lab 8. Then we will follow it with Lab 9. Lab 10 is shorter. It's very, very short, actually. Very short. So on Lab 10, I will do Lab 6 with you. We will set it up, I'll set up like maybe 4 microscopes for you guys to look into for Lab 6, and I won't make you do the whole thing, just to keep it an easy, nice, easy day. So we will come back to Lab 6. It will be with Lab 10, okay? And I'll change the dates when I can, but y'all please bear with me. I am not doing good. So just kind of be patient with me on that, okay? So FYI, while we're talking about Lab 10, and that'll be for everybody, okay? Lab 10 will be also Lab 6. All right, let's move on. Question 6, which of the following is not one of the stages in a PCR cycle? This requires us to kind of understand the terminology a little bit. Right, so good job. So we generally got this. It's ligation. We don't need to ligate anything in PCR. We do need to ligate things in DNA replication, but in PCR, it's such a short sequence. There's not going to be any ligating going on. So we don't need to ligate. We do need to denature and then anneal the primers and then extend the primers. And that's all we would need to do. We don't need to ligate anything. So good job. All right, question 7. What are plasmids made of? Let's see if we were paying attention. I've talked about them already. They are external from the chromosome. They are little circular bits of It's actually a pretty good picture of like the layout of a prokaryote or eukaryote. This is our bacteria, where we have our chromosome hanging out in the middle, and we have some ribosomes, and we have some plasmids. And plasmids are little circular bits of DNA. They are circles of DNA, and they are double-stranded DNA. Double-stranded DN. This is not working at all now. Circles. Wow, this is really lagging a lot. All right, so double-stranded DNA circles here. And they can contain extra genes that can help the bacteria, but sometimes it's harmful. If it's harmful, the bacteria will try to get rid of it. Some things that they can carry that can be helpful for the bacteria, they can carry genes for toxins that the bacteria can produce. Even genes for making a capsule can be on there. Genes for evading phagocytes could be on there. Genes for antibiotic resistance can be on there. And bacteria can shuttle this DNA to and from one another through various means. We learned about some of these means already, but I will kind of list them out. The first one is through, transduction. I don't know if I'm going to be able to do this. This is driving me insane. Transduction. stuck. Sorry. I don't know why this is like this. Let me see if I can type it faster than it is letting me do that. So this is the transfer of genes via bacteriophage and lysogeny. Remember that? So that whole process of transferring genes from the bacteriophage, incorporating it via lysogeny and getting new traits, which is basically lysogenic conversion of that bacteria, process of getting those traits by lysogeny is a process that we call transduction. I misspelled it. Transduction. Okay. The next one is transformation. This is by picking up, and only certain types of cells can do this, competent. We call them competent. Not all cells are competent. Not all cells can do this process. But when competent bacterial cells, it doesn't always have to be bacterial cells, let's just put competent cells, can pick up DNA from their environment. In lab 10, we're going to do a simulation of transformation. This is very commonly used in molecular biology to put genes into bacteria, okay? And then the last one is conjugation. No, not donjugation, conjugation. This is going to be a process by which bacteria create channels that we call sex pili to shuttle plasmids, usually plasmid copies, from one cell to the connected cell. Through the channel. The pili. Okay, so there's three types of ways that bacteria can transmit genetic information, and these methods are called horizontal gene transfer. And we'll talk about them again in more detail in lab 10 when we get to lab 10. Okay? It's way easier to do it that way than try to use my little thing that's lagging like nobody's business. Okay. All right, we're just going to finish up the Kahoot and then we'll be done. I know we're a little bit over time, but I really want to spend some time on this. If an organism contains any genetic information from another organism, what do we call it? This is more than one answer. Two of these mean the same thing. All right. So this is genetically modified. Yeah, sorry. Okay, so this is genetically modified, which means the same thing as transgenic. Is this unnatural? No, this can happen in nature. It's not unnatural. This can happen in nature. This is something that can happen in nature. Okay, so it's not necessarily unnatural. Does it always create a new species? Absolutely not. No. Okay. How do we define a species? That is a bigger discussion than we need to get into in my class. Okay. When you guys, if you take genetics ever, you can talk about it with your genetics professors. I'm not going to talk about it here. It's very complicated. All right, next. True or false? Some mutations in just one single nucleotide can cause devastating effects in organisms. True or false? Absolutely. So we even talked about this a little bit in the previous chapter with our different mutation types, that sometimes a single nucleotide change can cause something as simple as a stop codon, stop, which is a nonsense codon, right? And that can stop the production of the entire downstream from it. And that can be life-threatening. It can even cause changes in your amino acid. And that change in the amino acid can cause that protein to misfold and then no longer function. So think about when we're talking about mutations, don't just think about how it affects the DNA. Think about how it affects the amino acid, which affects the protein structure and therefore its function. And so that can be devastating to organisms. We call this phenomenon a single nucleotide polymorphism, or SNP. This means that you could have a nucleotide sequence that could be, AAA could get changed to AAU and code for a different amino acid. It could be UAU, and that could be a stop. And there's all these different versions of what we could see from just one nucleotide changing. And a single nucleotide polymorphism just means many versions. Some of those can be devastating. Sometimes they can be beneficial too, but they can be devastating if we change the amino acid or if we have a stop codon, which would be called a nonsense mutation, right? If it was a stop instead of what it was supposed to be. That makes sense? I hope it does. Then our last question, gene therapy. Tell me about it from our choices. Okay, so there's two major types of gene therapy. There's somatic gene therapy, which affects existing body cells. It can't be transmitted from parent to offspring. And then there is what we call germline gene therapy, which affects the gametes and can be inherited. It's heritable, which means it's passed from parent to offspring. Okay. Okay, well, this is just, I don't know why this is so laggy. Okay, so anyways, those are the different types of gene therapy that you can have. In this instance, gene therapy is literally treating a genetic disease. So if you have a genetic mutation, we can use HIV, for example. Most commonly, we use HIV. We remove any genes that could lead to HIV being HIV and instead replace it with working versions of the gene. So if you have a version of a gene that doesn't work, we can take your bone marrow out of your bones, infect it with our special HIV, which is not HIV anymore. You would not be HIV positive. You would not be HIV positive. You would not be HIV positive. It's not HIV anymore. We've changed its genes. Like if I take cells out of your body and I remove everything in it that makes it you, but I just still keep it cells, it's not you anymore just because it's cells. So it's the same thing with the HIV. Could this virus infect other people? Yes, but it would just give them a working copy of the gene. It wouldn't give them HIV. Okay. So we remove everything that makes it HIV and we put a working copy of the gene into somebody that doesn't have a working copy of the gene. They have a devastating disease, potentially, should be why we're using it. And we have at least 13 diseases now that can be treated this way, including, there's several versions of this, including like sickle cell disease can be treated this way. and certain clotting diseases can be treated this way. And there's neurodegenerative diseases. We really only use this as a last case situation because you can have negative side effects because we are giving you a viral infection and your body can still attack the virus, but it's not going to give you HIV. Okay. What it should do is give you a working version of the gene when you had a faulty one, a defective one. And it's the idea of how gene therapy works. Right now, we are usually treating people who are adults that already have a version of this gene that is wrong, and we're giving them a working version so now they can live a happy, normal life. Um, Whereas we are not usually treating, embryos or, gametes. And so the only way that this would be effective for it to be heritable and passed on to offspring is if we could change it in an embryo while it's still forming. So if we're doing IVF, for example, we could change the genetics of something while in vitro fertilization while it's still an embryo before we implant it. And of course, now you can start to imagine this would be a slippery slope. If we can give it a working copy of, a clotting factor, then we could give them blue eyes instead of brown eyes. We could make them taller. We could make them more, have a faster metabolism. We could make them more attractive. We could make designer babies, and that's a problem. So, FYI, so that would lead to something that we would call eugenics in the designing of people. And there's obviously a lot of ethics involved in that and how that would affect maybe people that aren't designed and things like that. So you haven't seen the movie Gattaca, and I would recommend watching Gattaca. It's Ethan Hawke and Uma Thurman, and they're great in it. But it explores that idea of basically designer babies and how it affects society. So anyways, that's it. Anybody have any questions for me on chapter 6 or 8? All right, if that's it, thank you guys for joining me today. That'll be it for today. Email me if you do have any questions. We're not going to go over the worksheets today. So if you are joining me for my Monday, Wednesday class, you will still have to work through the worksheets on your own. You're welcome to use other resources or work together on that to answer those questions if you need it. And you can always email me if you have any questions about the worksheets. And if you're here and you're like, what do you mean the worksheets? The ones that we usually go over in class, these worksheets. That you guys, if you're joining me in person and you're in an in-person situation, you don't have to do the worksheets and you'll get credit for this for my, Tuesday, Thursday class. If you've been assigned the hybrid version, you still have to do the worksheets. And these are those 5 questions that are associated with the case studies. If you want to work through these, you are welcome to. You can always find these. Just look for the traditional lecture application activity in your assignments. It'll be either at the very bottom of your assignments tab, or you can find it in your module, your weekly module. And you can feel free to do these, but if you're joining me in person, again, if you're not assigned the hybrid version, if you're not assigned the hybrid version, you automatically get credit for those for being here. And it's always been that way when we have these types of class. Okay, any questions before I let you guys go? We will have lab on Thursday. I may or may not be there. Just be on the lookout for that. But you will have lab and somebody will be there, and I will be available via Zoom. And so you guys will have the ability to ask questions, and I'll still lecture you over the content for Lab 7 on Thursday, Wednesday and Thursday for Lab 7. All right. Thank you guys so much. That's it for today. Um, yeah. That's it for today. I'll talk to you guys later. Yeah, Tuesday and Thursday, are we doing online lab for today? There's no lab for today. No lab for today. No lab 6. On Thursday, we will be doing lab. Is there in-person lab tomorrow for my Monday, Wednesday people? Yes. If you're on Monday, Wednesday, lab will be tomorrow. Yeah. Trying to check any other messages. I hope that's everything. All right. And you guys are welcome to leave. Yes, absolutely. All right. Thanks, guys. You do not need to do post lab 6. I'm going to move that to a different day. I mean, you're welcome to do it if you want to, but you don't have to. I'm going to move it. I just haven't had the time or the energy really to change it. So I will update the date on post lab 6. I'll actually do that right now. All right, thanks.